Thank you very much. Very good pronunciation. Difficult to pronounce name, but that was really good. Thank you very much. Thank you also very much for inviting me here to give this talk. It's really a pleasure for me to be here. It's the first time that I'm in this area, and it's a pleasure for me really to share what we generated in terms of research data over probably the last 15 to 20 years. I've been fortunate in the sense that my laboratory has received repeated funding on various aspects of physiology, fruit physiology, that all contributed to a better understanding of cherries. And what came out and what is our latest thinking about that process, I'm going to tell you in the next few minutes here. Now, as you can see, not only cherries crack, but also fingers crack. And if I'm using this as a pointer, please don't be offended. There are two fingers underneath here. So I have my talk structured into 3 chapters. Number 1 is going to tell you what everybody believes, why cherries crack, but I'm going to show you that that is not true. Number 2, I'm going to tell you a little bit about what we think that cherry... Say it again? Is that better? Oh, I see, okay. Okay, can you hear me? Good. Number 2 is I'm going to tell you our current understanding of why that process occurs. And number 3, I want to spend a little bit of time on what can be done about that. Now, I'm a research scientist, and I am not a Superman, and I'm not a magician. So when you go home, the fruit is still going to crack, okay? We start out with chapter number 1. This here is the student version, what you find in basically every horticultural book. You know, we got the fruit on the tree, the fruit is full of carbohydrates, it rains onto the fruit absorbs water through the skin, the fruit increases in volume, as the volume increases, the surface area increases, the pressure increases, and that is going to lead to fruit cracking. Now that is not true. I'm going to show you 4 reasons why that is not true. The first one is, if we wanted to balance the amount of carbohydrates that is in a fruit, we need to have really huge pressures inside the fruit. When we use that device here to measure the pressure, we realize that that pressure is way off, way too low to account for really an equilibration of the amount of carbohydrates that we have with the fruit turgor. So that can't be true. It's way too low. Second argument, I just showed you the story that as the volume increases, due to water uptake, the pressure should increase. Now on that x-axis we have water uptake, and on the y-axis we have the pressure. So that line should have a slope like this, but it does have a slope like that. And the fruit that is within that red circle is fruit that cracked. So what we can see is that even water uptake up to the point of cracking has basically no detectable effect on turgor. There is very, very low turgor in sweet cherries. Number 3, we can take a syringe and inject 2 mils of water into a 10-gram fruit. I used this fairly low fruit weight simply because it's easier to calculate. That means a 20% volume increase, and then the fruit cracks. If we put the fruit in water, it cracks with 1/10 of the volume. The story that I showed you before should produce equal cracking at equal amounts of water uptake, which it doesn't. Finally, if you take 2 test tubes, you fill one with water, and you put the fruit with the styler scar onto that test tube so that the styler scar is wetted, but the rest of the fruit is allowed to transpire. It's going to lose weight, but it's going to crack. So, in summary, cracking is not caused by high pressure in the fruit. That is a myth. Chapter number 2. Now here comes what we think is the true story. I need to expand a little bit and show you some microscopy images here. And what we see here is a cross-section through a cherry. The pit would be in the basement down there somewhere, and we take a look at the flesh. We see huge cells containing lots of carbohydrates and also quite a bit of malic acid. That is the organic acid that occurs at fairly high concentrations in cherries. We see as we move towards the outside, towards the skin, we see that cell size decreases and the the smallest cells we have in the outermost layer. And that is also where we have the thickest cell walls. So we have thin-walled cells beneath with lots of carbohydrate. We have small cells with thick cell walls containing much less carbohydrates, and the outermost layer produces something that I lined out here with the red line, and that you see that inset of here. And that is a biopolymer film that is produced by the epidermis and that is deposited on the outer surface. Now, these two things have very different functions. The skin cells, those are the thick-walled small cells there, they provide the structural strength. And the cuticle, which is that very thin biopolymer film, I forgot to mention that this one is only 1/1000th of a millimeter thick. So that very thin film on a mature fruit, that is the barrier that should prevent water penetration across the skin, also during processing. And it should also prevent invasion with pathogens. Now we need to take a look on how that skin develops. When our fruit grows, it increases in mass. And it does so particularly rapid during the second or during the third part of the development. When it increases in mass, it also increases in surface area. And you see that from 40 days onwards, we see, if you follow the red bullets, we see a very rapid increase in surface area as the fruit expands. Now the blue bullets show us what's happening with that polymer film that was responsible for the barrier properties. And you see from 40 days onwards, despite a 3 to 4-fold increase in surface area, there is no change in the amount of cuticle, in the amount of barrier for that fruit. So when the fruit turns from green to red, you see that mass increase and surface area increase. What's happening to the cuticle is what's happening to that balloon as it gets inflated. It still has the same amount of rubber, yeah, but it covers a much larger surface area, and that leads to stress, that leads to strain, that leads to tension. Unfortunately, it looks like that at least the cherry varieties that we looked at all have the same problem. They are all doing the same thing, namely nothing during the last phase of fruit development. And what I showed you here for the cuticle actually also holds for the cellulose that makes the major portion of the cell walls of the skin. That was the portion that was responsible for mechanical strength. Now that has tremendous consequences. When we take a look at a really nice looking, shiny sweet cherry, beautiful cherry, looks fine. But when we take the microscope, it turns out to look like a disaster, at least in certain areas of the fruit. Namely, the reason why we see a whole bunch of cracks, microscopic cracks that are too small to be seen with the naked eye. So that is something that is on the fruit, but we don't see it. And apparently, you see that these cracks, they are all oriented perpendicular to the longest dimension of the underlying cell. And why that is, I mean, you can easily guess from that belly there, you know, as the strain develops, tension comes, and then it cracks. And the crack is the result of the strain because these cracks are oriented perpendicular to the force that is generated. You see also, you can also try that out at home, take a razor blade, cut into the fruit, and you see that the fruit is going to gape, caused by tension. Now these microcracks, these microscopic cracks are entry holes where it is very easy for the water to penetrate into the fruit. It's going to penetrate into the skin, it's going to penetrate into the outer flesh. That is the place where most of the carbohydrates were. And then what we call a vicious circle is initiated. As these thick, thin-walled cells take up water, they begin to crack. They begin to lose water inside the outer flesh. And when they start cracking, they start to leak malic acid into the cell wall space. And if the first cell starts, then there is a little bit of malic acid, and the neighboring cells come into contact with malic acid, and that propagates the thing just like a chain reaction in a nuclear power plant. So we get a local defect that propagates due to the action of that acid. we made an interesting observation, notably that the cell walls, when they are exposed to this malic acid, that they thicken, they get very, very thick. And we were wondering, what does that mean in terms of cell wall mechanics? So we built a little device, and it's basically a pot filled with silicone oil, and on one part of the pot there is a lid, and that lid has a skin of a sweet cherry. So the pot is full with oil. Then we drive a piston into that pot. We displace volume, and where should the volume go? It presses underneath the skin so that the skin bulges. So we can measure two things. We can measure the amount of bulging, which represents an increase in surface area, but we can also measure the pressure. And to make a long story short, I summarized this figure down here. On the x-axis, we see how thick the cell walls are. And they are thick not because they have more cellulose. No, they are thick because these cell walls have taken up lots of water, and they were allowed to do so because the neighboring cells were damaged. Okay? So you see that as the cell walls become thick, the pressure required to destroy the skin became less and less and less until at the very end, we hardly need any pressure to destroy the skin. And there are a whole bunch of different treatments, and I'm not going to bore you with any details here, but there are a whole bunch of different treatments, and they all follow the same relationship. So the swelling as a result of the exposure to malic acid makes the skin weak, locally. Locally, because that only takes place, at least initially, where there was that microscopic crack. Now, when plant tissue ruptures, it can rupture differently. It could rupture across the cell walls. That is what that sketch above shows there. So we would have to rupture cellulosic cell walls, which requires lots of force. But the cherry doesn't rupture like that. The cherry ruptures like what I've encircled there. That is how natural fracturing occurs, and that is what we can reproduce with this apparatus. So the cherries fracture by separating adjacent cells. So what's happening is that the glue that glues cells together, that is a cell wall component called pectins, that glue becomes weakened, and it becomes weakened due to water absorption. I know that these are pectins because we have been using antibodies, and this is brand new. Nobody has ever seen that here. Greg, you should take a look. These are antibodies that recognize a certain class of pectins on the surface of these cracks, and everything that is green there is covered with pectins. And that emphasizes that really the separation is along those cell walls. So we came up with what we call the zipper model. You know all what a zipper is, right? The zipper model explains cracking, and it starts out with the tremendous increase in surface area without cuticle deposition. That causes the microcracks. We also get microcracking if we wet the surface. Just the presence of water on the surface generates microcracking, even if we prevented, for technical reasons, if we prevented water uptake. So that is something to keep also in mind. It makes things worse. That causes localized water uptake, and then we start with some individual cells that begin to crack. These ones release malic acid, causing the neighboring cells to become leaky. We get plasmolysis. I'm not going to go into that in detail, but we also get cell wall swelling, and that cell wall swelling makes the connection between adjacent cells weak. That decreases the fracture pressure, so the force required to propagate the crack, and the crack is going to propagate. This thing is going to spin until the fruit looks like what we see in that right picture. That is the zipper. So the skin gets unzipped in the same way like a ladder runs in a piece of knitted fabric. You know, on a piece of nylon, there it goes. So that's the way it cracks. Now the interesting thing comes, can we do something about cracking? Well, you will be disappointed what I have to say. I think part of the problem is that the breeders messed it up. No, I'm not critical about the breeders, but I think the breeders have a task to fulfill, and I think they can do a good job by trying to find genetic variation in the cuticle deposition pattern of the fruit. It would help if we found fruit that was able to form cuticles, still cuticle material, cutin and wax during late development. We haven't been able to do so, but I'm still hopeful that maybe somewhere we can find that material. What we learned from molecular biology is that the apparatus that synthesizes the genes are basically shut off. So it's not a lack of precursors that we could apply, but it's a shut off of the genes. The breeders could also develop an interest in cell wall swelling. I think cell wall swelling is very critical, and it depends on the type of carbohydrates that are present in the cell wall. And we have just started to understand that that is the problem. So now we need to look at what we have in terms of variation and how that changes. So I think in the long run that may help, but that's not going to help you that have to bring in a decent harvest this year. So what the growers can do are basically these 4 or these 3 green boxes. And I'm spending a few minutes on every one of them. Yes, it's a disappointment, but we have to reduce exposure to wetness. Basically, all the fruit cracks. There are obviously some that crack more than others, no doubt about that, and I assume that you are all paying attention to varietal differences, but they all basically crack. So rain covers, when they came out initially, everybody was saying, oh my God, this is going to kill me because it's way too expensive. We now know that it is still expensive, but it does work. It reduces it to a very significant extent. Some folks are using, at least in our countries, blowers. They drive around with crop protection equipment with no liquid, and then they blow off the fruit. Others even hire helicopters, but I think neither one of them has any indication of whether that strategy is effective. I think there is no decent scientific data on that, and whether it is economic. I think the helicopter is beyond discussion, but the blower, maybe that's not all too bad. Nobody thinks when they do that about the risk of bruising and pitting. The cherries do like this, and they bang against each other, so that is certainly going to cause bruising and pitting. And it's also going to loosen the pedicel, something that usually we don't want. We want fruit with pedicels. There's a fraction that thinks that wax coatings are very useful. I know these commercial folders, and these folks show up in regular intervals at the farms, particularly during the harvest period. But I think, again, effectiveness is not proven. There is no scientific, sound experimentation that proves that this stuff works. One of the problems is coverage. That cherry has been hanging on a wash line. And we have been spraying that from all sides, so you can't get a better canopy architecture than a free-hanging cherry on a wash line. And when you measure the amount of coverage that you get, you have about 18% of the surface covered. The remaining 82% is going to be uncovered, not treated. And I'm going to promise that will have no effect on water transport. Also, the question about selectivity. We have heard about photosynthesis. Our leaves need to gas exchange. And if we provide a shield around the fruit, how are we going to manage to provide that shield only for the fruit? And then the fruit can't respire. We will also coat the leaves. And the leaves require gas exchange. So these things, I think, they're not going to work. So that leaves really the covering. Problem number 2, and I think that is something that may turn out to be more promising. You all know about calcium. Calcium has beneficial effects on fruit quality, and calcium also is cross-linking cell wall constituents. So basically, calcium has 2 arms, and it can grab onto one thing in the cell wall, and it can grab onto the other thing and hold the things together. And indeed, when we do so, we find better cell adhesion, and we find reduced cell wall swelling due to calcium. Yes. And cell wall swelling was critical. The trouble with calcium is, and I'm showing you that with a few images there will be very little calcium uptake into a mature fruit via the vascular system. When we feed a dye through the pedicel, you see only the red color in the immature fruit, but you don't see any color in the mature fruit. And the reason is because the mature fruit has no functional xylem anymore. It ruptures. And xylem is one part of the vascular system, and the only part where calcium can be translocated. So there will be no import via the root and the trunk, at maturity, and it will be continuously decreasing during early development until it is basically shut off. So the only way when we get it via the root system into the fruit is during early development, and that means we need to maximize transpiration during early development, because it is transpiration of the fruit that drives calcium transport. We have very nice relationships between the amount of fruit transpired and the calcium uptake, but that can be only done during early fruit development, and there is obviously an interaction between canopy architecture, that was the story that Greg was telling, and calcium uptake. There's lots of variability. If the cherries are occluded, they contain very little calcium, but if they are exposed, they probably do more. If we want to improve the calcium supply later, we have to come with multiple sprays. And we just did an experiment, I think it was last year, where we applied 6 times concentrations, and I think we used calcium chloride, calcium formiate, and I think these two, at concentrations that are very close to leaf burn. So it's probably the maximum load that we could take. And we really banged it. So we get a 67% increase in calcium uptake, but we were really disappointed because the effect on cracking was fairly low. The time for 50% of the fruit to crack increased from 2.6 hours when we sprayed water to 3.9 hours. That is even less than doubling. And the total percentage of cracking was not affected. But I think there is still hope. So I think performance could be improved if we were able to get higher calcium concentrations into the skin, particularly when these cracks start to developing. What is unlikely to be helpful? I don't believe in irrigation scheduling, at least not in mature fruit. And the reason is simply because the xylem is not functional. There will be no direct controllable response of water supply to the root system on changes in fruit diameter. I don't think that that's going to happen. The story is different in immature fruit. There, the xylem is still functional, and there I think we may be able to do something. What is also not going to happen and not going to work is spray application of osmolytes. You know, in the past, people have been using sugar solutions to spray the fruit in order to reverse the osmotic gradient that drives uptake through the skin. We will only produce sticky fruit, and it will not work because when we apply it, it's going to be water soluble, and when it rains, you know, the water solubility takes care of wash off. I discussed film-forming agents, and I don't believe in those. There are magic products, silver bullets, or snake oils that may help, and if it helps the wrong people. Yeah? What actually may help is go to church, light a candle, and do some prayers, or take your wife out for dinner. No, I really think the calcium story has some potential, but I don't believe in these things. I would like to thank you very much for listening to me, and obviously the stuff that I am presenting has not been produced by me alone, but I had a whole bunch of people helping me doing this kind of research. Thank you very much.